All right, so a reminder here about our lab final, which will come up on April the 20th. We couldn't get this room, so we got the room on the other end over there, 127. Laid out the same way. So you'll go in and set, spread out where the says set here. And it's at 6:30 until 8:30 of the normal 2-hour time. It probably will not take that long, but you have it. And that will be for all 3 labs, no matter which day you have lab, we're all have, so everybody can take it at the same time. And then it'll be done with lab. So I wanted to go over some of those problems that we had, I had listed from chapter 9 today. So I need a blank slide now. So the first one was to look at some codons and anticodons and match them up. So we'll go through those. And this was problem number 7. And it had several different ones. The first one, they're all 5' 3', was UAG. And this is the anticodon that you're being given. So the question is, what amino acid would be carried by the tRNA that has this codon? So you would look at your codon chart and you would look for the codon of the amino acid that goes with the codon. So when they base pair, they're going to base pair anti-parallel. So the codon that would be here would be C, U, and it could be A, or it could be G. Because U, this is the first position of the codon, second position, wobble position. Okay, so the wobble is the third base of the codon. So which base is it of the anticodon? Actually, the first nucleotide in the anticodon. So that's the wobble position. And remember that U can base pair with A, or it can base pair with G. So if you look up that codon in your codon table or your codon chart, CU anything is a leucine. So this should be leucine. Letter B was, the anticodon was AAA. So the codon would be UUU, and A doesn't have any wobbles. So actually, I don't know that there is an AAA anticodon. In fact, I don't think that that's even real, but if it was, you would be limited to UUU, which is phenylalanine, as we learned yesterday with our genetic code. And there is no alternatives. The other one, the next one, C, was CUC. Okay, so C can base pair with G, U can base pair with A, C can only base pair with G. So GAG should be glutamic acid. Right, so this one was leucine, that one, that's an L, this is an F, this is an E, if you want the single letters. There's not going to be an alternative because C doesn't have an alternative wobble. Okay, D had the sequence A, U, G, and you're going to, oh, it's a start codon. Remember, this is the anticodon, so it base pairs with C, A, and U because A doesn't have a wobble. So CAU corresponds to the amino acid histidine. All right, our last one was the anticodon GAU, which would base pair with an AU. And then A can base pair with C, or it could base pair with U. So you have two possibilities, AUU or AUC, which I think is an isoleucine. So the next question said, look at them and look at both potential codons that it could base pair with. So I did that already in that answer. So the only two that have alternatives are actually A and E. The E, C, and B, C, and D don't really have alternatives. Okay. Now, if you got some weird thing, you'd say, oh, well, but A can base pair with inosine, but inosine is never going to be in the mRNA. Okay. So the weird bases, okay, the weird bases like inosine, not in a codon because it's not in the mRNA. It could only be in an anticodon. Okay, so the weird modified ones can only end up in an anticodon or in a tRNA. And honestly, there aren't any for humans. All right, so that answers questions 7 and 8, really. The next question was question 12, which showed you this picture of a ribosome and an mRNA and it being translated and said there's some problems. There's a lot of problems, so you're supposed to list them all. So try to point some of these out. First one, the ends are wrong. If translation is happening this direction, it translates from the 5' end to the 3' end. So the first codon that would have come into the ribosome would now be this codon that's in the E site, which is TTC, which is wrong because it's got thymine in the RNA. So this should be the 5' end, this should be the 3' end, this should be uracils there instead of T's. The codon UUC, what does it code for? Look that up for me right quick. UUC, huh?
Phenylalanine.
Phenylalanine, okay? So the very first amino acid in this protein or in this thing should have been phenylalanine, but of course phenylalanine wouldn't be the first amino acid, so something's wrong, we're missing amino acids, right? And AAU wouldn't have been a start codon, so we're missing amino acids, but that's okay. So if it had have been UUC, then phenylalanine needs to be here because alanine just got attached, right? So phenylalanine should be the first one, and then GCA, what is that the codon for? Alanine. So alanine and phenylalanine are in the wrong places. And here you have CGT as the anticodon, and that couldn't be, so that needs to be a uracil. Okay, you have the codon CCG. What does that code for? Not glycine. I'll tell you that for sure. Proline. So that's wrong. That should have been proline there instead of glycine. And coming into that A site. So there's some mistakes. What other mistakes can you spot? Huge glaring numerical mistakes.
Should be 50 and 30 S.
Well, it's this eukaryote, right? Does it say it's a eukaryote? Eukaryotic ribosome. So this is a 60S. and this is 40S for eukaryotes, right? And the whole thing is 80. Is the whole thing. So those were the problems I found. There may be actually others, I don't know. Not showing you the start codon, so that means you're missing amino acids, but I don't know if they meant to do that or not. All right, so if you can redraw that and make it right, you can understand many of the features that need to be going on during translation. A new slide, another new slide. All right, so problem 17 said that there's a line below, which isn't below, it's to the right, but there's a line here representing a eukaryotic messenger RNA, a mature eukaryotic messenger mRNA, and they've listed some things that they want you to draw or indicate where those things would be, and they tell you some of them might not be there or aren't supposed to be there, okay? I list them out. So let's draw our line here, and we're going to have to add to it here in a second. So I'm going to draw it in the middle. So here's our line. So the first thing, a stop codon. Yeah, that should be on there somewhere. I'm not going to put anything on until I know what all is supposed to be on there. A poly A tail. That should be on an mRNA, a mature one. An intron. No, that should not be in a mature mRNA. That's removed by splicing. A 3' untranslated region. Yeah, that would be there, the area after the stop codon. A promoter. No, that's never in the mRNA. A start codon. Yeah, we should have one of those. AAU, AAA, which is what? What is that sequence? Poly A signal, the polyadenylation signal. It's not the tail. The tail is all A's. This occurs 13 bases prior to the poly A tail. A 5' untranslated region. Yes, it should have that. It should have a cap. Yes. It should have a termination sequence. No, not an mRNA. Transcription has to terminate, but the mRNA is just ends. So where all these things go, I'm going to start with the cap because I know where it goes. It goes right there at the 5' end. And then somewhere there has to be a start codon, and it's going to be closer to the 5' end. So I'm going to put it there. Start. So everything from here to there is 5' UTR. So now I know where that is. Then somewhere down here, I'm going to have a stop codon. I'm going to put it right there. And everything after that is there. The poly A tail would be on the end. So you could either draw A's here. I'm just going to draw it hanging off the end further. So that's my poly A tail. And 13 nucleotides before that, okay, is my polyadenylation signal, AAU, A. So I do a little thing there to say, okay, it's right around here. And everything here, technically only up to the beginning of the poly A tail, but if you included the poly A tail, I don't think anyone would hold that against you. I wouldn't, but I don't, but I wouldn't fault you. All right, so that 3' untranslated region is everything after the stop codon, okay? So we left a few things out, like introns are not supposed to be in there. I don't want to put it up there. Intron, the promoter. Did we leave anything else out? Termination sequence. And these are things that would be used to make the RNA, but aren't part of the RNA. So then question 18 says, all right, you did that right. Now, draw where the protein goes and line it up. So here's our protein. So it's going to line up with the start and the stop codons, right? The region between these start and stop, what do we call it? It has a name or an acronym. The open reading frame, right? So for our protein, the start codon and the 5' end of the RNA correspond to the amino terminus, and the stop codon and the 3' end of it correspond to the carboxy terminus. So it said label those termini on the peptide. Then it said carefully draw 2 lines. I didn't follow the instructions. That's totally threw caution to the wind. Above and parallel to the mRNA, and label them coding or template. Well, by tradition, the top strand is the coding strand. So the bottom strand has to be the template strand. And then it said on that strand, find all the parts. So locate the promoter sequence and the +1. Well, the +1 needs to line up with the first nucleotide here of your RNA. Not the cap, but the first nucleotide. And you have the AAA, except it's AAT, signal here. And you also have a signal, oops, just after that, that is a GU-rich signal sequence. And together, those are the termination signals for a eukaryote. It terminates and cuts between them where I drew the dotted line. This up here is your promoter. What would you have in there? There would be a TATA box, right? Around -25. So you could throw that on there. Okay, I think that's everything it asked for. Now, if this was a gene that had introns and exons, you would also have, it wouldn't be perfectly the same length. The DNA would be longer, but it didn't say to do anything about that, because I think it didn't want you to know that introns shouldn't be in the RNA. But we could say that there are, let's say that this is an exon right here, and then there's another exon down here, and we'll just say this thing has 2 exons. It actually goes to the cut site. So they got spliced together, like right there, boom, boom. And so that corresponds to our mRNA, like that, if you want to. And this could be an intron right here. Okay. What was next? 26. 26, I can write it up there. It says you had 146 amino acid protein and beta globin. So how many codons would it take? How many nucleotides would it take? Right? So you have to think about it. Each amino acid corresponds to a codon. So that's 146 codons. Each codon is 3 nucleotides. So you got to multiply that by 3. So that's 438. But then you need 3 more because of the stop codon. So take the number of amino acids times 3 plus 3, and you always have the number of codons in your reading frame. So actually it would take 441 codons, nucleotides to encode that. So the stop codon, remember, doesn't correspond to an amino acid, but you had to know it, it had to be there, it had to be used to stop the whole thing, right? To get exactly 146. So it's important, and it has to be counted. It's necessary. All right, and then 30 was the last one. I think I put that on here. So this one was a tricky one, okay? So I won't trick you like this, but they're trying to be sneaky. So they said locate this DNA encodes 5 amino acid polypeptides. A 5 amino acid polypeptide. So first you have to find the start codon and then the correct codons after that. So you have to look at the 2 strands. So if it's written as it's supposed to be written, that's 5 prime, that's 3 prime. So if you look at the whole first top strand, you do not find an ATG anywhere. So it can't be the coding strand. So now you come on the second strand, but you don't start on this end because you have to read it 5 prime to 3 prime. So you have to go backwards and look for an ATG. There's one. It can't just be any ATG. It needs to be an ATG in a Kozak consensus. ATG G, that's really important that the next nucleotide be a G. And if we go before it, it should be a purine. And that is not a purine, but it's the only one that works because there's not another ATG that will give us 5 amino acids. So we're stuck with that one, even though it's not a good Kozak consensus, we have to live with it. So that's tricky. They gave you something that's not a good consensus, not like what I gave you on the quiz, which was going the right direction, and it had a absolute perfect match to Kozak consensus. It was the third ATG over, by the way. So ATG, so there's amino acid 1, right? Methionine, and then the next codon would be GTA. What's that in code? Look that up. You'll be looking for U on your chart, but same thing. Who? Valine. And then CGG. You can see what we're going to do here, so you can go ahead and look ahead. DCT or UCU. You're all looking, so anyone is fine to call it out. Serine?
Yes.
GAT?
Aspartic acid.
TAG?
Stop codon.
So that's our 5, isn't it? MVRSD. I wish they'd made it something fun. Like what happens if you have the amino sequence glutamic acid, leucine, valine, isoleucine, serine? Yeah. So some British dudes sitting around spending some time on the computer searched for that 5 amino acid sequence in the protein database at the time. This was back when I was in grad school in the 90s. And they found it. So you could theoretically predict using statistics, knowing amino acid probabilities, like there's 1 out of 20, but it's not really 1/20th because some amino acids have more codons, right? Leucine shows up more often, serine shows up more often than, say, glutamic acid. So you put in all of the perfect matches for human DNA for how likely ELVIS should appear in protein sequences just by randomness, and then they search for it. And it comes out higher, not just a little bit, but way higher than statistically predicted. Their conclusion was Elvis is alive, and he's in all of us, in our proteins. So that's that single letter abbreviation. So this would mean that our coding strand is this strand, right? And our little amino acids would be going backwards there. What else did they tell us to do? Give the sequence and polarity of the mRNA encoding the polypeptide. So the mRNA sequence would have been A, U, G, G, U, A, right? Just switch all the T's for U's. Yeehaw, you can do that. Give the polypeptide sequence, we did it. So the amino terminus would be here, and the C terminus would be here. And that gets a little scary when you're using single letter abbreviations because that is an amino acid, and so is that. Assuming the sequence is a bacterial gene, identify the region encoding the Shine-Dalgarno sequence. Well, it's right here, AGG, AGG. There's our Shine-Dalgarno box, because AGG is a beautiful Shine-Dalgarno box, but that's pretty rare, but a couple of A's and a couple of G's together usually makes a Shine-Dalgarno box. And what is the function of that?
Ribosome binding.
Yep. So the ribosome, the 16S subunit of the ribosome can base pair with that, and it positions the rest of the ribosome right there so that the initiator tRNA can come right there with its F-met on it and be in the P site, right?
So the Shine-Dalgarno box is not always gonna exactly be GGAG.
No.
Okay.
Yeah, so I probably want to ask you to identify one since there's not a beautiful consensus for it. And it's also not really perfectly placed either. It's pretty variable. So kudos to them for actually coming up with something that they could find, because a computer, you had to actually, the first one would have had to have been found by eye because the computer would never have done it. They can make it, make an algorithm now that'll do them, but it's not a really good consensus. That was all of them, right? Yeah, I think so. Okay, so we want to spend the balance of the time here today talking a little bit about gene regulation, not just in bacteria, although we'll start in on bacteria with details, but in general. So first off, we think about gene regulation, and we've talked about the term gene expression, meaning a gene and its information being used, right? So a gene will be transcribed, and genes make RNA, encode RNA. Could be ribosomal RNA, could be tRNA, could be small nuclear RNAs. There's a lot of different kinds of RNAs out there. Could be a messenger RNA, right? If it's a messenger RNA, then it's going to go on to be translated. So the final product would be a protein. But the regulation of building that final product, either it being the RNA product or a protein product, is what we're talking about here. How does the RNA polymerase know where to start? Well, we know it has to recognize promoter sequences. We know in bacteria, the RNA polymerase itself has the ability to bind to the -10 and -35, but in eukaryotes, the RNA polymerase doesn't really bind to any specific sequence. It needs to be grabbed by other proteins that bind to DNA sequences. So the regulation between prokaryotes and eukaryotes, it has a lot of similarities in mechanics and in principles, but some major differences in detail. We have the thought of genes that are expressed and needed and used all the time during the life of a cell, or in every cell in an organism even. So there's two things to think about. When is the gene expressed? Constitutive expression means that it's expressed in constant on. The gene is just on. It's being used all the time. And this would include things like the ribosomal RNA genes. There's never a time when a cell really doesn't need a ribosome unless it's going to be a red blood cell. And our red blood cells, in fact, build all the hemoglobin they can and a few other enzymes that are in them, and then they shut down all of that process. They eliminate their ribosomes, they eliminate their mitochondria, they eliminate their nuclei, and all they are is a bag of hemoglobin and a few other enzymes for mammals. Now, other organisms don't do that. So birds and reptiles and amphibians, their blood cells are still nucleated and they still can transcribe, and they actually live longer than our red blood cells. So our red blood cells are dying the day that they leave the bone marrow and are mature. And they're going to last about 120 days, which is pretty good for an organism, for a cell. Think of a cell as an organism with no brain, because its nucleus is gone, right? It has no machinery to repair itself with. So it's pretty good. But every other cell has to have ribosomes, has to always be translating, has to make tRNAs all the time. Plus, there's a whole lot of genes like sodium, potassium pumps, and all of the catabolic and anabolic proteins that are used to build stuff and get energy from the nutrients the cell is taking in. All of those things have to be made all the time. And so there are basically genes that are constitutively expressed. Well, that's a no-brainer. You got a switch, you throw the switch, you walk away, you forget the switch forever. It's just always on. But then we think about the genes that make tissues different, cell types be different, and do specific tasks in our body. Those genes in those cells that are doing that specific task should only be on there and then, and not in other cells. So when we talk about things that aren't constitutively expressed, we're going to refer to them as differentially expressed. It's like the reverse of that, differentially expressed. Okay, and so that differential expression could mean that it's in different cells, and it can also mean that it's at different times. Okay, now the other term is, where is the gene expressed? The other concept to think about. Ubiquitous means everywhere. Okay, it's expressed in all cell types if you have more than one cell type, or it's expressed during all stages of all life cycles that the organism may have. Okay, so when we think about this type of expression, again, kind of the opposite to this is differentially expressed. It's not just expressed everywhere, it's expressed in different places. And this is where we can get all Star Trek on it. Temporal and spatial. Temporal, different times. Spatial, different places. So genes expressed at different times. What do we mean by times? Well, say before you got infected with a virus versus after you got infected with a virus. That's temporal difference. When you were a 3-day-old embryo versus when you were a 70-day-old embryo. Okay, so developmental temporal thing. Spatial differences in the liver versus in the kidney, right? That's a spatial difference. So temporal and spatially expressed differences of differential expression have to be regulated. There has to be a mechanism that says, okay, now is the time, get turned on. Now is the place, be active. Now is not the place, get turned off. Now is not the time, right? So things have to be both turned on or turned off as necessary to build the organism the way the organism needs to be built, or the way it works. So we can really think about expression as being differential or not differential. And if it's not differential, then it could be both constitutive and ubiquitous, or it could just be constitutive at this place, or ubiquitous and at different times and things like that. All right, so we have already talked about transcription, and we said that, well, starting the process is where the lion's share of regulation is going to be, and that's true. So if you don't ever have to transcribe the gene, you don't have to worry about any of the other stuff. Splicing, translating, none of that stuff has to happen. So regulating when and where transcription starts is what most regulation of gene expression is all about. So we have in our sort of broad picture of regulation, there are two categories of factors that can modulate it. You have factors that make it stop or keep it from going. Those are repressors. You have factors that make it start or make it go more. Those are activators. So you basically have a dichotomous thing going on with the nature, repressors or activators. These things usually are proteins which bind directly to DNA. They probably also interact with other proteins like the transcription apparatus itself, the RNA polymerase or basal machinery, and they may sterically block it, they may physically hold it, they may physically help it bind, they may physically stimulate its activity. So they can do different things chemically or biochemically to how they work. But in general, they bring about either repression or activation of a gene. Okay? In bacteria, these types of proteins are a little different in the way they can function than in eukaryotes, because in bacteria, sometimes these things can be coupled with the translation process to a degree. Since there's no nucleus and those events are happening simultaneously, you can have a little interplay there. In eukaryotes, you don't get to have that. So there's a few things that eukaryotes can't do, ways they can't regulate their genes, yet there's ways they can that bacteria can't. So it's kind of a trade-off. Okay, if a gene is controlled by basically keeping it off, then we say its regulation is negative control. It needs a repressor to make it not be transcribed. And in the absence of that repressor, it would be transcribed. So its default state, we think about its default state. What would RNA polymerase be doing with this gene all by itself? Well, it would be transcribing it. So how are we going to keep RNA polymerase from doing that when it's not supposed to? Well, we're going to have to repress it. We're going to have to probably get in the way. That's how most of them work. They sterically prevent transcription. Okay. What if the gene, if left up to its own things, RNA polymerase totally ignored it, then that gene would be inherently not transcribed. So it would be off. How are you going to get it turned on? Well, that's where a protein like an activator would be necessary to first bind the DNA and help RNA polymerase get working. So that gene's default state would essentially be off, and to make it run, you have to actively turn it on. That's known as positive control, right? So you have couch potato genes that you have to come along and say, oh, okay, get up and go do something. And you have those people that are, let's go here, let's go here, let's go here, let's go here. And you're like, okay, dude, I'm tired already. And you're supposed to be asleep right now, right? So you kind of got both things going on with your genes. And so they have to be regulated in opposite ways. Now, truth be told, most genes do not only use one thing. So they may inherently be off, but they may not just come on either. You might have to keep them off, but you might also have to say, go ahead and start whenever the repression is relieved. So most genes have some level of negative control and positive control, both, that can modulate their level, right? So in a lot of transcriptional regulation, when a gene needs to turn on or be turned off is going to be in response to environmental factors, right? Like you got infected with a virus. So how do you know you got infected with a virus? Well, your immune system cells that are responding to the virus can release chemicals like cytokines, right? And that's one of the problems with COVID, the cytokine storm, where your immune system basically signals too much and the immune system starts to cause damage to your tissues over and above what the virus has done. So those kinds of environmental signals, they could be cell signaling molecules like hormones, cytokines, or they could be chemicals from the environment. So for like some bacteria, you know, if they sense a nutrient and they can sense that there's a gradient to it, right? Because they have receptors that are picking up that nutrient. Well, it will activate genes that will cause them to proliferate or to migrate in the direction of that gradient. So these external cues, external to the cell, have to be picked up, and that can modulate transcription. So these would be called effector molecules, right? They are affecting either turning genes on or turning genes off. The molecule, right, it's just a chemical. It doesn't bind to the DNA. It usually binds to some sort of a protein. We call them proteins receptors or sometimes it binds to a repressor, sometimes it binds to an activator, right? And it can make that protein work. So any small effector molecule that increases or enhances the amount of transcription is known as an inducer. Okay, and the way an inducer works is by inhibition of a repressor. Okay? So if a gene was wanting to be on, let me be transcribed, and there was a repressor going no, and then an inhibitor, an inducer comes along, it binds to the repressor. Now the repressor can't repress, so you have transcription, right? So I call it my cool thing in nature. A cool thing in nature. It's not mine, it's ours, it's nature's. But inhibition of inhibitors. It happens way more often than you normally would think, right? Occam's razor is simplest answer should be the simplest solution should be the answer, right? And inhibiting an inhibitor is not does not follow Occam's razor thing. So, but that's the way nature works a lot of the time. Okay, I want that gene to be on, so I'll just turn it on. Well, you don't. You actually have to turn off the thing turning it off so it can come on. There are things called coactivators. They cause the gene to be expressed because they interact with an activator who wasn't actually activating yet and cause the activator to actually function as an activator. So anything that needs another molecule to bind to it to make it do its job is an apo thing, right? So you would have like an apo activator, apo. So it's not working. And then along comes an activator or co-activator because it's activating the activator. And now you have the co-activator and the activator working, functioning, right? So apo means it's a thing that needs something else to go with it and it doesn't have its other thing yet. So you have apo repressors and apo activators that can function. So a co-activator binds to an apo activator and activates it to be an activator. Can we say activator one more time? You got the idea though? So any gene that works like that, if an inducer causes it to be turned on by blocking a repressor, or a co-activator turns it on by activating an activator, then it's called inducible. Induce means to turn on, cause to happen. What about the flip side? Things that make transcription be turned off or down. Co-repressor. So you have a repressor, but it's not actually a good repressor. It's not really wanting to repress. So it's really an apo repressor. So you have a co-repressor, which is a small molecule of some kind that binds to the aporepressor and turns the aporepressor into a bona fide repressor, which would then block transcription. What if you have an activator? It's a good activator. It's activating all by itself, but you need it not to activate right now. So you actually need to inhibit the activator. And we actually call these things inhibitors. So an inhibitor would bind a functional activator and turn off its function, stop its activation function, and so turn DNA transcription off. Genes that need this to happen are known as repressible genes. So we'll look at two examples. We'll look at an operon in bacteria that is considered an inducible operon. So it's a situation where it needs to be turned on. Its default state is essentially on, but it's repressed, so it has to be induced. And then we'll look at another one whose default state is off, or should be off, and has to be turned on when it's needed. But the way it's kept off is by constant repression. So one is kept off by lack of transcription, poor transcription, and the other one is kept off by active repression. All right, so this makes sense. A lot of little things. The book has pictures of all these things. Yeehaw! You can look at those pictures, and you can see what they're trying to tell you there. So, to some of the people who worked out this stuff did it a long time ago by your calendars in the 1950s and late 40s. Jacques Monod and Francois Jacob. Jacques over there, this is Francois, and they worked with this guy also, Andre Wolf, and they worked in France, and they actually did some of this stuff during World War II, and that was their day job, and their night job was actually they were part of the Partisan. They were out there blowing up train tracks and disrupting the Germans. So not all scientists are just boring science labby guys. They weren't making bioweapons. They were just literally blowing up people, but not people, but trains and stuff like that. One of them got blown up himself. I can't remember which one it was. He had shrapnel in his butt. So, yeehaw. So here's what Monod had noticed. He had noticed that bacteria, when you give them food, they have different preferences for different food, so what's food for a bacterium, right? Good juicy steak, just different carbon sources in their media. And he also noticed that if you gave them two foods at the same time, they tended to use up all of the one food before the other. And their preferred food of all was glucose, right? So glucose was their steak and potatoes. Or if you're a vegan or vegetarian, not steak and potatoes, just the potatoes and the soy. So whatever, they had glucose. Glucose is, the 6-carbon sugar that cells like to use, so it makes sense. If you gave them another sugar at the same time, they would eat all the glucose first and use it up, and then they would not grow for a little bit, and then they would decide, okay, we're still hungry, let's eat the lactose, right? It's like Coke versus Pepsi, although now you can't, Dr. Pepper versus anything else, right? So you're like, yeah, Dr. Pepper first, and then whatever's left, right? Toilet water, then Coke, then Pepsi. So why, he was just curious about, so why didn't they just use them all at the same time, A? And why, when they ran out of one, did it take them a little while to start using the second one? And so he said, well, I'm going to look to see if there's any different proteins that have to get made. And so he did electrophoresis, and he found that there was another protein that shows up after they're able to start eating the lactose. And he figured, well, that protein probably allows them to eat the lactose. So I'm going to call that protein beta galactosidase. And he turned out to be right, although it was kind of a guess. And so he could look for the presence of that protein as one of their diagnostic tools. And then he got to talking with his buddy Francois down the hall, and Francois had been doing stuff with viruses, and he saw the same kind of thing. Like you infect the cell, but then the virus just sits there dormant, and then you have to do something to the cell to make the virus actually lyse the cell. He was working with lysogens and inducing the lysogens, and he used the term induction, and that's where the term induction came from, and inducing things. And so together they said, well, let's do stuff. Let's look for mutations that don't do this. Let's look for ones that use the lactose all the time, eat the lactose right at the beginning, can't eat the lactose at all, don't pause in between, pause forever, you know, things that go wrong with the normal. So there was normal. Oh, what am I doing? There's normal. So anything that's not normal, that would be just eating everything all at once. That would be not pausing, that kind of thing. So he said, okay, sounds good. So they started looking for these things, and they found groups of mutations, and they called them inducer mutations. And they named that mutation LAC-I for induction mutation. Long story short, so they mapped out a variety of mutations, and the mutations fell into several different regions next to each other on the chromosome. They mapped out mutations that they found that couldn't regulate the process, and those mutations fell here in what we know now is the LAC-I gene, which encodes a repressor. They also fell into sequences between the LAC-I gene and the beginning of the LAC-Z gene. And they named those mutations operators because they seem to control the whole process. And the term operator heralds back to that age and day when you didn't just call somebody, you called the operator. So you pick up your phone, it connected to a telephone office where there was an operator who then you said, connect me to Bob, and they took a cord and they plugged your cord into their cord, and now you were talking to Bob and them, because the operators could listen. And I know because my mom was an operator, did exactly that. She knew everything in town. This operator, they called it, was in control. It really let things be connected. And then, of course, you had the enzyme beta-galactosidase itself, and you had a couple of other reading frames that seemed to encode different proteins that they couldn't identify them, but they could note that they could make antibodies against it, and it was turned on and turned off. They couldn't see the protein. So they said, okay, we kind of know where things are. Here's a more blown up version of that. And you can see on here that the repressor gene, LAC-I, its reading frame stops there. And then there's a sequence that we'll come back to where a protein binds. Then there's the -10 and the -35 where the RNA polymerase binds. And then there's another sequence here that is the same as the sequence over here and the same as the sequence that's down here as well, but they didn't show it to you. And that's the operator sequence. And it's just at the beginning of the start codon, or the start site of transcription. And then here's where the protein starts actually over here, and here's your Schindel-Garno box, AGGA, and so on and so forth. So all the parts are there, you can look at the sequence. So this promoter, this operon, is regulated in two ways. There's a repressor that binds to the DNA and blocks transcription, and that repressor is made by the LAC-I gene, and that gene's promoter is constitutive. Not very good, but good enough. So there's always a little bit of repressor protein being made, a trickle all the time. It's not a drip, it's a leaky faucet that's got a little drizzle coming down from it, okay? That repressor binds to this operator sequence here, and it binds to the operator sequence back here. Those are its two favorite sequences. And I did this, they didn't show it, but if you read this sequence, G, C, A, G, T, G, A, blah, 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 and you read this sequence, they're very similar. They're not identical, but they're similar. I'll let you do that on your own time when you're bored. But that is where the repressor binds. It binds to two places simultaneously. But the repressor is not two repressor molecules doing that, it's one giant tetramer doing it. So it actually makes those two spots come together like this, which means all this stuff in the middle turns into a loop. And that keeps transcription from happening. Now, if the repressor is released from the DNA, transcription should occur, right? Theoretically, it can. This is not a strong promoter. It's got a couple of mismatches, like it has GT right here instead of AA, and that turns out to be kind of important. So it doesn't really bind to RNA polymerase very well. So it can be turned on when the repressor is missing, but it isn't very much. It's not a drizzle, it's a very slow drip, okay? So how do we turn it onto a full stream? That's where the cap protein will help. So this sequence right here in purple, the repressor will be gone. Now this sequence can be bound by a protein called cap. Cap responds to a co-activator called cyclic AMP. When would you come into contact with cyclic AMP? Cyclic AMP gets made when the cell is getting low on energy. So when it's getting low on energy, it needs to eat any other source of food in its environment, any other sugar. So that cyclic AMP activates a protein called CAP, cyclic AMP activated protein, binds to the DNA and will cause transcription. Now, if the repressor is still involved, it can't. But if lactose is around, lactose causes the repressor to fall off. So if lactose is available, you could eat it if you need to eat it. Okay, so that's why they saw the curve. Lactose is around the entire time, but so is glucose. So lactose is around, the repressor is gone, but glucose is present, no cyclic AMP. The promoter is not on. It could be on, but it's not on. Now they run out of glucose. Now the cyclic AMP spikes. Now CAP is activated. Lactose is around. Eat lactose, turn on the lac operon. Okay, so it's a two-part regulation. Part one, stay off unless lactose is here. Part two, turn on if lactose is here and eat it. Okay, if lactose is the only thing that's there, right? So you have negative control with the lac repressor encoded by the lac-I gene. You have positive control caused by the activator protein CAP. Now, CAP also goes by the acronym CRP. CRP stands for cyclic AMP response protein, and CAP actually stands for catabolite activated protein, but the catabolite is like lactose or something else. What they really know now is that it's cyclic AMP is activating it, so people will call it cyclic AMP activated protein, and they'll call it CRP, and I don't know why they haven't combined those acronyms. What would that be? Crap, right? So anyway, they haven't. I think our book uses CAP. Other books, other people might use CRP. It just depends on the person. And so you have, like I said, it has to have it's a two-part thing. You have to get rid of the repressor and turn on the activator. It's kind of a double control on it, right? Don't just come on because you're hungry. Only come on if you're hungry and this food is here. because it costs you energy to make the protein. If you make the proteins to eat lactose and lactose isn't the food, you just wasted more energy when you're already running low. That's kind of a good, kind of a elegant system. All right, we'll talk a little bit more about that and eukaryotic ones on Tuesday. And I guess we'll have no quiz on Tuesday. We'll be quizzing on Thursdays, I guess now. So that'll be fine.